Good morning and warm welcome to everyone present. We begin this gathering with Tamil Thai Vazhuthu. May I request everyone to kindly rise. Thank you. Kindly be seated. Respected Honorable Chairman, Medical Director, Dean, distinguished scientists, faculty members, researchers, students, and honored guests. It is a privilege to welcome you to the 6th World Microbiome Day Annual Lecture 2026. To formally welcome the gathering, I invite Dr. S. Mohanraj, Scientist, KMCH Research Foundation, to deliver the welcome address.
Thank you, Brahmi. அனைவருக்கும் காலை வணக்கம். கற்றது கைமண் அளவு, கல்லாதது உலக அளவு. What we have learned is only a handful. What remains to be learned is as vast as a world. This is beautiful quote remains as
off பண்ணிட்டு on பண்ணிட்டோம்.
That science is endless to learning the discovery and the experience. Good morning everyone. It gives me immense pleasure to welcome you for the 6th World Microbiome Day celebration organized by KMCH Research Foundation. Microbiome research has become one of the most exciting and rapidly growing area in biomedical sciences. Today, we know that trillions of microorganisms living in and on our body play a vital role in maintaining our health. They influence our metabolism, immunity, digestion, mental health, and also the development of metabolic diseases and even chronic conditions. World Microbiome Day is celebrated every year to create awareness about the importance of the microbiome and to encourage the scientific research and collaboration in this fascinating field. I would like to extend a special welcome to our chairman, Dr. Nalaji Palaniswamy, our medical director, Dr. Murugan, our director of KMCH Research Foundation, Dr. Arulraj Ramakrishnan, and the director of KMCH IS, sir, Dr. Kalanidhi, sir, and Vice Associate Dean, Dr. Sridhar, sir. We are truly honored to have an eminent professor with us today, Dr. Silpi Sharma, IIT from Delhi. Dr. Sharma has made significant contribution to microbiome research, and we are privileged to have the opportunity to learn her experience and expertise. On behalf of everyone at the KMCH Research Foundation, I extend to warm welcome you, madam. I also take this opportunity to warmly welcome to our respected dignitaries and Dr. NGP College of Faculties and students, clinicians and researchers who have joined us today. Your presence makes this event more meaningful and encourage scientific discussion and collaborations. I sincerely thank our chairman and management for their continuous encouragement for the support of research and academic activities. I hope today's session will inspire all today explore the remarkable world of the microbiome and its potential of transform the future of research. Once again, I extend the warm welcome to our distinguished guests and each one of you. I thank you and I wish you all wonderful, enriching World Microbiome Day celebration. Thank you.
Thank you, sir, for your warm words of welcome. The liver is the body's great chemist and the hepatologist is the greatest advocate. To present the activities and achievements of KMCH Research Foundation, I invite Dr. Arulraj Ramakrishnan, President of KMCH Research Foundation and consultant hepatologist, whose contributions to healthcare and research continue to inspire excellence and innovation.
Thank you. அனைவருக்கும் வணக்கம். ஸோ இந்த ரிசர்ச் ஃபவுண்டேஷன் பற்றி பேசணும் அப்படின்னா வந்துட்டு ரொம்ப சிம்பிளாக ஒரே வார்த்தையில் முடிச்சிடலாம். நல்லாம்பட்டியில் ஆரம்பித்து அனைவரும் நலம் பெற அப்படிங்கிறது தான் இதோடைய கான்செப்ட்டு. ஸோ வாட் வி ஆல் ரியலைஸ், ஒரு மேற்கத்திய நாடுகளில் ட்ரெயின் ஆகும்போது, வெஸ்டர்ன் வேர்ல்டில் இருக்கும்போது, வாட் வி ஆல் தாட் தட் வாஸ் இட் இஸ் தேர் ஆக்டிவிட்டி, எவ்ரி ரிசர்ச் ஆக்டிவிட்டி வாஸ் ட்ரைங் டு சி சிட்டிங் இன் அன் ஐவரி டவர். அதாவது ஒரு கண்ணாடி மாளிகையில் உட்காந்துட்டு நாம் வந்துட்டு ஒரு சட்டம் போட்டு மக்களுக்கு நல்லது பண்ணோம் அப்படிங்கிறத இருக்க முடியாது. தரையில் இறங்கி வேலை செஞ்சோன்னா தான் மக்களுக்கு என்ன பிரச்சனை இருக்குது அப்படின்னு. ஸோ சேம் திங் இந்த KMCH ஹாஸ்பிட்டல் அப்படிங்கிறது நோய் வந்ததுக்கப்புறம் அவங்களுக்கு ஒரு நல்ல ஒரு குவாலிட்டி, ஹை குவாலிட்டி கேர் அப்படிங்கிறத எஸ்டாப்ளிஷ் பண்ணோம் அப்படிங்கிறதுக்காக KMCH ஆரம்பிக்கப்பட்டது. இந்த KMCH ரிசர்ச் ஃபவுண்டேஷன் வருமுன் காப்போம் திட்டம் தான். நோய் வர்றதுக்கு முன்னாடியே மக்களுக்கு எப்படி என்ன ஏது அப்படிங்கிறது. ஸோ தட்ஸ் வாட் வி வாண்டட் டு. வி ஸ்டார்டட் அ ஜேர்னி. 2014 we started a journey. ஸோ அந்த ஜேர்னி ஆரம்பிக்கும் போது சிம்பிள் திங், வி வாண்டட் டு ஹாவ் அ சார்ட் ஆஃப் an idea about what are the diseases in our part of India, and after that how could we change it. So we've been doing 10 years study. ஸோ அந்த 10 இயர்ஸில் நாங்கள் பார்த்தது என்ன அப்படின்னா மக்களுக்கு இந்த மாற்றங்கள் வர்றதுக்கு பல காரணங்கள் இருக்குது. அதை புரிஞ்சுக்கிட்டு ஒவ்வொன்றும் we started implementing in our labs and see that whether we could get an answer. So let's say for example, நாங்கள் மண்ணில் பார்த்தது நம்ம யூஸ் பண்ணக்கூடிய ரசாயனம் ஃபெர்டிலைசர்னால மண் மாற்றம் வருது. So we started looking it in our lab, did sort of work on that to see that if you were to give the same minerals to the rats and see that whether we could produce the same diseases. So சிம்பிள் உதாரணம், கிராமத்தில் இருக்கவங்கள உழைப்பாளி, அவங்களுக்கெல்லாம் சர்க்கரை வரக்கூடாது அப்படிங்கிறது தான் எல்லாத்துடைய எண்ணமும் கூட. பட் வாட் வி ஃபவுண்ட் வாஸ் இட் இஸ் ஈவன் த ஃபார்மர்ஸ் ஹேவ் காட் ஹை ரிஸ்க் ஆஃப் டயபிட்டிஸ். ஸோ த ரீசன் ஃபார் தட் வாட் வி ஃபவுண்ட் வாஸ் இட் வாஸ் ப்ராப்ளி த ஃபெர்டிலைசர்ஸ் that what we're using, and as well as the pesticides and insecticides what we're using. So we did the same experiment in our lab. We gave the fertilizers and the insecticides to the mice, and we found that the mice which weren't given fertilizers or pesticides didn't develop diabetes. On the other hand, the mice which were given pesticides and fertilizers did develop diabetes. So bingo, we got an answer. மக்களுக்கு பிரச்சனை இருக்கிறதுக்கு காரணம் என்ன? So we wrote a grant to the government to say that this is what is happening, and we're in the process of being distributed the grant, the concept being one health. And the one health அப்படிங்கிறது நம்ம மனிதகுலம் மட்டும் இல்லாமல் நம்ம மண், நம்மளுடைய உயிர் நண்பர்களாகிய ஆடு, மாடு, பன்றி, கோழி எல்லாத்துக்கும். ஸோ தட் ஒன் ஹெல்த் இன்வால்வ்ஸ் எவ்ரி ஒன். ஸோ தட் கிராண்ட் இஸ் பீயிங் டிஸ்ட்ரிபியூட்டட் ஆஸ் வி ஸ்பீக். ஸோ வி ட்ரைங் டு சீ தட் நம்ம லேபில் பார்த்ததை மக்களுக்கு ஒரு நன்மையாக மண் வளத்தை முன்னேற்றம் கொண்டு வருது, நீர் வளத்தை முன்னேற்றம் கொண்டு வருது, அதனால் நோயை தடுக்க முடியுமா. ஸோ தட்ஸ் the journey. What started us defining the diseases has gone down to see that how we can change so that people don't get the disease at all. So அதனால் அனைவரும் நலம் பெற அப்படின்னு ஆரம்பித்தது நோய் வருமுன் காப்போமா இன்றைக்கி வந்துருச்சு. So that's how the journey of KMCH Research Foundation over the last 12 years have been. I'm not going to blow a trumpet about the number of publications what we've done and all those things, but the most important thing what we would define as a journey would be the contribution to health for this nation. We probably would be coming out with a document in this year, would be our more than 10 years of our celebration of KMCH Research Foundation to say that how much we contributed to the health of the nation, and also the stakeholders can take some information from it to make some changes. So thank you everyone, and we hope that our organization has been enriched by several speakers, and of that the fifth speaker, we've been really humbled by acceptance of the invitation by all the speakers from worldwide. So we've had many distinguished speakers, and today we got another distinguished speaker who's going to tell you about microbiomes and soil, which is where our research has been. I hope you all have a great day. Thank you very much.
Thank you, sir. Visionary leadership and enduring dedication to healthcare, education, and social well-being form the cornerstone of every great institution. With immense respect and privilege, I invite our Honorable Chairman, Dr. G. Nalla Palaniswamy, a distinguished leader whose commitment to excellence has elevated our institution to greater heights, to deliver the presidential address.
Good morning to you all. Today, I know you are all from our colleges, and you may not know English or any other language other than Tamil. We combine the English and Tamil together. Anyway, எல்லாத்துக்கும் வணக்கங்கள். இன்னைக்கு 6th World Microbiome Day. For that, we have a very great personality from Delhi, IIT Delhi. She is here with us, and she is going to talk all about the microorganisms, which benefits the human beings. There are many microorganisms. It's not good to the mankind, but many organisms good to have it in with us, so that it helps us have a nice life in the planet Earth. That is what all about it. And microbiology, microorganisms are so many organisms in the air, in the water, and also in the soil. These organisms are very much needed for our life. Without that microorganism, we cannot live. That type of situation we are having it. And we are very proud that we have a good team of people who are in the KMCH Research Foundation. For this, our respected Professor Silpi Sharma is here. I hope she will give us some information about what is the microbiology and what the organism is going to help, especially soil organization. I heard she is very much interested in soil organization. What are the soil organisms which are going to help you? And human being, we have from the air, from mouth to the end of the body, you are all having so many bacteria. Those bacteria are very helpful, and we have a great professor with us, Dr. Chokkavelu from West Virginia University, and he is going to also talk about something about the microbiology. And you will have some information about the microbiology and how this microbiology or microorganisms are help to you. And so many, I don't know whether she is going to talk about the other than bacilli or there are so many microorganisms. There are so many microorganisms from the virus, from the fungus, and also from other minute organisms you may not know. Those are the organisms are in the planet Earth. This weather, the temperature and water and oxygen gives these organisms to grow and live. That we are very, very proud that we have a very big, knowledgeable people who can give you some information. The students, especially I like to inform you, you have to have eager to learn about what is happening in the earth and how these organisms are helping us. This is a curiosity. That curiosity only developed so many vaccines all over the world. These organisms are very important for the life. Dr. Chokavelli is going to introduce, one of the person going to introduce the HIV vaccines from United States, and that type of senior person here with us. And I am very proud that our KMCH Research Foundation, these people may not know what is a KMCH Research Foundation. This basic of KMCH Research Foundation is, especially students I am telling you, there are infectious diseases for attacking the human beings. There are non-infectious diseases we have in India, everywhere in the world. Especially, we did not have the proper guidance or information, the statistic of how many non-infectious diseases are available, prevalence in India. The KMCH Research Foundation took that challenge. What is the prevalence of the non-infectious diseases? Today we are all talking about infectious diseases. And while we are talking about KMCH Research Foundation, mainly established for to see what is happening in our place, whether infectious or non-infectious. We are very much interested. They are doing good job, especially waterborne infection, how that waterborne infection is helping the agriculture. Especially there are so many irrigation places, the lakes, rivers, and all canals. There are so many things, there are so many sources of the water pollutions, and we may get some information about if you study about the waterborne infection. Our KMCH Research Foundation involved in that also. We are very proud that they are very much eager to learn about what is happening. Long time ago, there is a study in America, what is happening in these places, that is called Massachusetts, and in the state of Massachusetts, there is a study called a Framingham study. You all go and see your Google or whatever you have on your cell phone, what is the Framingham study is. On the Framingham study basis, we have the Research Foundation. We are all doing, they are doing Dr. Mohan Raj, Dr. Arul Raj, all are doing good job. We also, why we are all here is, you should also some enthusiasm and interest about what is all about the research is, what is we can do all about is, you can involve with the research areas also. What is research is to find out new things. Whatever it is not there, you are going to find out new things. Whatever it is, even they talk about the soil, you can talk about the air, you can whatever it is, well, even human beings. That is, whatever it is, if it is not there, it is not found, you can have a curiosity to learn about what is happening in your studies. And you should know some kind of curiosity. The curiosity only, you become a scientist. And you all have to be in that type of situation. Madam, we have tons of PhDs. In India, there are tons of PhDs. Your higher education always says, how many PhDs in your college? What is that mean about the PhDs? I do not know, madam. Only degrees now. I am all talking about the PhDs are only not research oriented. They become degrees, madam. When you are in Delhi, talk about what you can do about, you should be really research persons, research oriented person. It's not a degree oriented PhDs, madam. I am very proud that for this, our Professor Silpi Sharma is here and he is going to talk about it. And now I request, before madam, I will ask Dr. Chokkavelu to talk about why, then your speech, madam. Okay. Dr. Chokkavelu. Okay. All right, madam.
Thank you, sir. It is now privileged to felicitate our distinguished guest speaker, Professor Shilpi Sharma, as a token of our respect and appreciation. May I request our Honorable Chairman, sir, to kindly felicitate Professor Sharma with a shawl and memento. I please request all the delegates to occupy the front row. Now I request Dr. G. Velmurugan, scientist, KMCH Research Foundation, to give a presentation about outreach and community engagement of KMCH Research Foundation.
Good morning all. So before going into the lecture, I just want to give a very short talk on what we are doing beyond science. Okay, so hope Dr. Arul Raj and Chairman Sir has detailedly explained on our activities. So these are our major course and vision focus. So our science doesn't end just in the laboratory, and it is not fulfilled with the publications alone. So this is a statement by a famous science communicator called Sagan. So we live in a society exclusively dependent on science and technology, in which hardly a few knows anything about science and technology. Even in our everyday life, so many things we have started utilizing. It is not science and technology is beyond our reach. But how these are happening, it's not reaches everyone. Just like every day we see a lot of political news or other developments and things. But how these are being developed, there is a lag. And we have took this mission, so we are committed on bridging this gap between science and society by communicating research in accessible language, engaging communities, nurturing scientific curiosity, and translating evidence into meaningful action for public benefit. So these are some of our activities. So we communicate this in our studies have been communicated for the public in news and print media. So these are some of our publication newses, and we are proud to say one of our recent study has been appeared as a news article in the famous Science magazine in the issue of April 2026. And also some of our studies on pesticide and gut microbiota has been incorporated in the UNFAO policy documents. So community engagement, as explained by everyone, our studies started from the communities. We go to the fields, we interact with the people, and today in all these 3 places where we are doing epidemiological studies, it's like relationships to us. We have become part of these villages. We know everyone there. We are, I would say, எனக்கும் நல்லாம்பட்டி ஒரு சொந்த ஊர்னே சொல்லலாம். அந்த அளவுக்கு it has become very bonding for us. So these are the 3 villages, and we go as a big team of people. We do all this health screening and activities. And also we have did a large scale study all over Tamil Nadu also in 2020, where we have engaged with nearly 61 villages all over Tamil Nadu among 0.1 million people. So this is a study funded by ICMR, led by Dr. Thomas Alexander and Mohan Raj. So I am proud to say, as part of this study, we saved a few lives. Okay, people came for screening, and we have advised you are at risk, and in some cases, they ignored our advice, but in the next few days or few months, they have developed cardiac problems. So we are proud to say, beyond publishing in Lancet Regional Health, we have saved a few lives. So, and also we are in a mission of creating awareness programs also. So now ICMR has, Dr. Mohan Raj has a grant on studying the prevalence of fatty liver among children. So it is not stopped with just the screening and programs. We create awareness, we go to the schools, we interact with the parents and the children, and we provide what is a fatty liver, what kind of lifestyle modifications are needed, what would be the future, because the results are alarming. We are seeing a huge prevalence of these things. And also obviously we work on microbiome. So as Chairman said, not all the microbes are bad. So we spreading on these messages. We have also created a video on microbiome in Tamil, which is in YouTube, and over the last few years it has reached nearly 10,000 students in this. And also in collaboration with IIT Madras, we are involved in enhancing the water literacy. We are part of an NPTEL course jointly conducted by IIT Madras and Tel Aviv University. So far we have trained nearly 12,000 students all over the country, and every student will collect data also. And also I am proud to say here, Dr. NGP College is the topmost institution by contributing to this. So we have nearly 800 students from Dr. NGP College, and you can see, so this is made up open data source. So there is a dashboard called People's Water Data. So all the water quality data is made freely available. And as a continuation of this, we have some real-time sensors implemented in the field, and we are now have a joint project with IIT Madras, which we are waiting for funds, where we will have all the data available, and people will have in the mobile, they can see the water and soil quality data, and some decision-making tools will be made. So this is the plan. I'm not going into details of this. So we will have a lab also in Alambatti village, which will be the native home of Chairman. Thank you, Chairman, for this. So, and also we involved in capacity building. So Professor Supavan Tantayan, who is the president of Federation of Asian Chemical Societies, has visited, and we have conducted a small scale workshop for the school teachers to enhance the learning among teachers and the school students. So these are some of the images. And also we are interested in creative communication. Last year we conducted this workshop on SciComm tunes. So these are some of the cartoons made about microbiota by the participants of that workshop. So I will skip these things. So and also, as a reward for all this, recently we got awarded an outreach grant from India Bioscience, where the theme is we have to communicate about One Health for the school children through science comics. So thanks to Dr. Kannan and Sabarish, who are the creators of these cartoons, where we have 2 characters, Tina and Luna. So Tina is a small girl, and Luna is a hypothetical flying squirrel. So where in the comics, every time Tina will ask about something and Luna will give scientific explanation. And so for example, this is about saprophytes. So we have started doing this activity, and few series have been done, and altogether this will turn out into a book. And so we will continue to translate, communicate, and disseminate science in ways that inspire society, empower communities, inform policy, and improve lives. And thank you, one and all. Thank you.
Thank you, Sam, for highlighting the remarkable initiatives and contribution of KMCH Research Foundation. Now, I call upon Dr. Sabarishwari, National Postdoctoral Fellow, to introduce today's chief guest, Professor Shilpi Sharma.
Good morning, everyone. Let me start with a quote on this beautiful occasions. A woman in science does more than require knowledge. She inspires generation to believe that curiosity has no boundaries. It's my great privilege and honor to introduce our first women speaker for today World Microbiome Day annual lecture by Professor Dr. Shilpi Sharma. And Professor Sharma is currently the Associate Dean of Academics and Outreach New Initiatives and the batch of 1980 Chair Professor in the Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. And she has been associated with Delhi from 2008, progressing from assistant professor to professor through her outstanding contribution to research, teaching, and academic leadership. She earned her BSc Honors and MSc Botany from Banaras Hindu University, where she graduated with multiple gold medals and university distinctions. She completed her PhD from Ludwig Maximilians University, Munich, Germany, under the guidance of Anton Hartmann, and subsequently she worked as a postdoctoral research scientist at GSF National Research Center for Environmental and Health in Munich, Germany. Professor Sharma is internationally recognized for pioneering research on microbial ecology, plant microbiome interactions, rhizosphere microbiology, climate resilient agriculture, and One Health approaches. Her research focuses on harnessing beneficial microorganisms and soil microbiome to improve agricultural sustainability, crop productivity, and environmental health. Her excellence has been recognized through numerous prestigious awards and honors, including Tata Transformation Prize in Food Security and Professor B.N. Jory Award from Microbial Diversity and Women Scientist Medal of Indian Society and the Women Impact Leadership Award from IIT Delhi and the Young Scientist Award from the Asian PGPR Society, as well as the award from the Environmentalist Association of India and the Association of Microbiologists of India. She has recently been elected as a Fellow of Indian National Science Academy and the Fellow of National Academy of Science, India. So reflecting her significant contributions to science and the society, passionate about teaching and research, she has been authored more than 120 research publications with holding patents and has successfully contributed to technology transfer. Beyond her research accomplishments, Professor Sharma actively serves as a scientific community through various national and international professional society and editorial boards. She has been mentored numerous students, researchers, and has been long-lasting contribution to advanced microbiome science in India and globally. Today, she will deliver on the lecture entitled The Microbiome Revolution Towards Sustainable Agriculture on One Health. Please join us in this extending and warm welcome to Professor Dr. Shilpi Sharma.
So, Vanakkam KMCH. I have to admit that's the only Tamil word that I know. Thank you all so very much. Thanks, Sabarish, for this nice introduction. Believe me or not, I'm really nervous today. There are two reasons actually. Number one being the stalwarts that have actually been speakers before me in the last 5 years. And the second reason is because I'm talking to healthcare specialists and I'm talking about One Health. So it's a great initiative talking about the microbiome, creating awareness about the microbiome, what is the might of the microbiome, and celebrating this on the World Microbiome Day, which was on the 27th of June. I have titled my talk as the Microbiome Revolution, and I will focus on sustainable agriculture. What is the role of the microbiome in sustainable agriculture and the One Health? I come from the Department of Biochemical Engineering and Biotechnology, and I thought I'll briefly introduce you to the department, which is one of the first departments to have recognized the importance of bringing together engineers and biologists. And as of now, we have 4 programs in the department. One is the UG BTech program, the MTech program, MS Research, and PhD. The USP of the department, as I mentioned, is that we are equal number of faculties who are engineers and biologists, and we try to solve some of the real life issues based on the engineering principles. Now, what I have been trying to do in the first decade of the formative years, I was trying to understand what are the different factors which affect the microbiome, which determine the composition of the microbiome. And the systems that I've been working on include, of course, the plant microbiome interactions, but I've also been working towards understanding the interaction between microbiomes and fabrics and the biological wastewater treatment system. So we're trying to understand or maybe predict the performance of a wastewater treatment system before it actually turns foul. And in the last few years, I have been trying to manipulate these ecosystems, the microbiome compositions, and engineer the microbiome for various ecosystem services, for various biotechnological applications, thereby trying to directly address some of these SDGs that you see listed here, but there are several others which are indirectly also being worked upon. This is how I've structured my talk. I'll first talk very briefly because I see there's so many students here. I'll try to take you all through the basic definition of what microbiome is, just so that we are on the same page. And then I'll talk about something wherein we all will relate. We'll talk about the human microbiome, which is also called as the invisible organ. I will then move to the plant microbiome. I'll try to show to you how there's a perfect analogy between the human microbiome and the plant microbiome. And finally, I'll talk about how the microbiome actually serves as a unifying factor amongst all different ecosystems. And in all these stories, I'll mention a bit of what I have been doing into research. I'll not boggle you with a lot of numbers. Okay, so what is the microbiome? There have been several definitions, and the definitions have been evolving, but simply put, microbiome is microbiota plus the theater of activity. When I say microbiota, I'm talking about the living component, the bacteria, the archaea, the fungi, protists, and so many other microorganisms. But then when I say the term microbiome, I'm actually also encompassing a theater of activity. Here it means the structural elements, all the elements which are released, several metabolites, and the environmental conditions. So that is how a microbiome is defined. And a great friend of mine, Gabriela Berg, and this paper, I would strongly recommend you to go through this paper if you want to see how this term has evolved. Simply put, the microbiome is the orchestra. And when I say that, the microbiota can be compared to all the musicians in an orchestra. Okay? They are the ones playing the music or would play the music. And so that's a taxonomical gradation. That is who is present in what numbers. That is more taxonomical, right? When you talk about metagenome, a word that is very commonly used, you're talking about all the music that the musicians can play. That is the potential that they have. But when I talk of microbiome, I'm talking about the actual orchestra that is being played, which is actually influenced by several factors, including the response of the audience. So this is a beautiful analogy that I found on the net, and I thought I'll present to you. After having told you about what the microbiome is in the current concept, let me talk about the human microbiome, which, as I said, is also the invisible organ. Till some years back, it was believed that the bacterial cells or the microbial cells greatly outnumber the human cells, now we know that we are equally, if not more, microbes than human. So you will see that the number of human cells and the number of microbial cells on our body, within our body and on our body are almost equal. In fact, this has led to us rethinking about the definition of what an individual or an organism is. We are really, typically a biologist would some years back say that a host is mostly the individual, the human, right? But now we're thinking about how the self is blurred with the non-self, because several microorganisms are closely associated with the humans and in fact impact the regular metabolism, help train the immune cells and perform several actions in an indispensable manner. We are never alone. We actually have, the humans have a core blueprint, but then there is a lot of expansion of activity possible because of the hundreds of microbial genes that are associated with the human, the self. And in fact, the humans and the microbiomes have been co-evolving. So basically, the microorganisms have been training the immune system to identify between what is beneficial and what is harmful. So much so that this co-evolution and the co-evolution has actually changed with the lifestyle. You will see that earlier what was a foraging diet had changed into more domestication and agriculture coming into picture. You will see that there was a change in the microbiome as well, right? And now we have a more industrial diet, which is processed foods and lower fiber, thereby leading to what is called as the old friends hypothesis. Now I mentioned that with co-evolution, the microbiome was actually training the immune system to identify between the beneficial and the harmful. Now with the sanitized, more sanitized environment, such kind of a training does not take place, and so there has been dysregulated immunity, increased allergy and inflammation reports. There is a perfect stratification. There is microbiome zoning in our body, and this is both macro zoning and micro zoning. So you have different kinds of microorganisms occupying different parts of our body, but even within the tissues you will find a zoning. And the maximum diversity and the total counts are found in the gut, with some of the areas like the stomach and the duodenum having more acid tolerant microorganisms. They are more aerated, while the distal colon, which has less aeration, has a lot of nutrient rich substrates, harbors the maximum number of microorganisms. And these of course have roles to play in producing vitamins, increasing immunity, and several other functions. You've heard about the gut-brain axis. We now have increased publications to talk about the microbiome actually modulating the gut-brain axis. In fact, there are papers talking about how the microbiome can actually impact the neurophysiology as well, the behavior as well. Now, how do we acquire the microbiome? This is a paper that I found, a recent paper in 2023 that came up, wherein they were comparing the fraction and how we would acquire microorganisms. And it's been seen that in the early life, it is majorly the mother's transmission through the mother that happens, while in the later part of the life, it is the other factors, including your environment, that plays a role in the microorganisms that we acquire. So they were comparing actually twins and several sets of individuals, and they found, these are the fractions that they found in terms of transmission of microbiome that was happening. The unique you, it's not just the fingerprint. In fact, microorganisms and the microbiome have been considered to be the biological barcodes. What I'm showing you here are 4 individuals with different lifestyles and the different environments that they are exposed to, and you will see that this kind of uniqueness is not just at the level of species, it goes down to the level of strains. That's how different you all are. And when, I'm sure you've heard about this, you've heard about transmission through physical contact, so whether it is the phone, whether it is the elevator, the coffee pot, whenever you touch something, you're actually transmitting microorganisms. But some studies have also shown that the physical contact is not all that transmits microorganisms. So for example, this paper by Meadow et al. In 2015 talked about we emitting microorganisms as you sit here, as we walk or as we move, up to a radius of approximately 90 centimeters. So now in this room, when you actually step out, you will be taking some mementos from your neighboring fellow people in the form of some bacteria or microorganisms. And these, called as the personal microbial cloud that we emit, are actually again signatures of individuals. So if I say they are unique for each individual, you can understand that they could have an application in forensic sciences as well. Now there's several areas in the forensics where the microbiomes have already been used. For example, studying the geolocation, what is the kind of substrate and the microbiome in the substrate that is present. You could have people localizing through the animal microbiome. There are several applications in forensics that have also been applied. There's a thumb rule. When there is a balanced microbiome, an individual is healthy. But there's something called as dysbiosis, and the microbiome misbehaves for several factors. There could be several supplements, there could be several environmental factors, which actually lead to a compromised diversity or dysbiosis. Now, dysbiosis has three markers. Number one, loss of diversity. Number two, loss of beneficial microorganisms. Number three, increased proportion of harmful bacteria. So this is what is called as dysbiosis in any context, whether it is the human microbiome, the plant microbiome, or the environmental microbiome. If that is the case, the idea is to replenish the good bacteria, feed your bugs, okay? You and your microbiome are what you eat. Now, there have been several studies wherein there have been, microbiome have been used from the bench to the bedside. You will see there are several types of therapeutics, microbiome therapeutics, including the fecal microbiota transplantation, which involves transplantation of the fecal from the healthy individual to an unhealthy individual. Microbiome has also been used for diagnostics because I told you they're so unique to every individual. However, there are several challenges to application of microbiome research in clinical practice. Of course, the biological variability is one of the reasons, but there are also certain logistical constraints, so challenges associated with the practical implementation of microbiome profiling because of the variations that you have. There cannot be a universal method per se, and the limited confidence of most clinicians in dealing with microbiome science also prevents the clinical application of the research data. So what can we do to have this transition from research to clinical practice? While there are several reasons that have been, or several points that can be worked upon, I wish to mention that the trial design needs to involve a lot of stakeholders from different areas, and also the communication. We need to establish connections between clinicians and the microbiome scientists. Now talking a bit about what I have done in the area, and this was together with my colleagues from the Department of Textile and Fiber Engineering at IIT Delhi, Professor Deepti Gupta and Dr. Avinash Sharma from NCCS Pune. As medical care people, we all know that hospitals serve as breeding grounds for hospital-acquired infections. What we were interested in is how some of the soft surfaces like fabrics actually serve as vectors in transmitting diseases. Now there's a problem of transfer, the transfer of infection from healthcare workers to patients. There's also transfer between patients. You will see that there's transmission also to the visitor who's coming, and most of the work that has been done or was done when we started was with hard surfaces like bedside railings and several other surfaces. What we found was there were a number of papers indicating how important the fabric is in these transmissions. What we knew when we started was that textiles do act as good substrates for adhesion, growth, and transmission of microorganisms. The interaction between the fabrics and the pathogens increase the risk of HAIs or nosocomial infections.
The uniform of healthcare workers actually served as fomites for transmission of antimicrobial resistance. Several kinds of multi-drug resistant bacteria and fungi were found. And the environment between the bed sheets and the patients were actually conducive for bacterial proliferation and was linked to the nosocomial infection. Now the solution here that was propagated was that antimicrobial coatings can be used, which could be antibacterial, antifungal, antiviral. However, they have their own challenges like environmental pollution, like it would not differentiate between the actual good skin microbiome. And so, what we found missing was that there was a lack of understanding of bacterial adhesion on soft surfaces. More work was done on hard surfaces. And what are the factors which affect this adhesion? There were hardly any studies in the Indian hospitals and healthcare units, and I already told you that the environment really played an important role in such kind of a transmission. There were no studies using culture-independent methods. When I say culture-independent methods, less than 10% of microorganisms can be cultured. So most of the studies were based on culturing the microorganisms. And there was limited study on mapping the antimicrobial resistant bacteria present in such kind of a setting. So what we did was, we tried to understand the impact of the fabric type and bacterial surface properties on bacterial adhesion. After having studied adhesion, we went on to transmission. We tried to understand what are the different factors which lead to transmission of the microbes, especially the pathogens. And finally, after performing all these conditions under in vitro labs environment, we went to the real life scenario. We characterized the bacterial community dynamics on fabrics along a time course as a response to seasonal variation. And we also mapped the antibiotic resistance profile on these fabrics. Now, like I said, I'm not going to boggle you with data. I'm just trying to tell you how we summarize this. We found that the adhesion of the microorganisms was favored by the nano roughness of the fibers. It was also affected by the surface hydrophobicity of the bacteria. The type of bacteria was also impacting it. When we looked at transmission, we saw that moisture between the donor and recipient was actually favoring the transmission. And dynamic friction, we were comparing static friction and dynamic friction. When dynamic friction was applied, it was more encouraging transmission of microorganisms. When we went on site and performed our studies in a primary healthcare unit, we were comparing different fabric types. So we were actually comparing nurses' lab coats, we compared pillows, bed sheets and curtains and the kind of fabrics that are used there. And in fact, what we did was we performed some patch experiments like this. So we would actually have different kinds of fabrics stitched onto the lab coats and then sample them after different shifts. And in this case, like I said, we studied the microbiome dynamics on fabrics and healthcare unit. We tried to see how the shifts were impacting the microbiome load. We also studied how antimicrobial resistance was actually impacted by seasonal variation. We saw what all microbial genera are actually present on these fabrics and tried to also identify which are the potential pathogens on these fabrics. Now, what are we currently working on in this area? We're trying to see how biofilm formation on the fabrics and how this can be disrupted. We're trying to see how different bodily fluids affect the fabric microbiome and how different appendages of the microorganisms affect the adhesion. Okay, after having told you something about the human microbiome which you could directly relate to, let me now move to the plant microbiome, which we believe are the drivers for the next green revolution. Students, there's a perfect analogy between all what I told you about the human microbiome and the plant microbiome, especially the plant roots and the rhizosphere, the zone surrounding the roots, which is completely analogous to the gut. How is it analogous? Both the systems are open systems with large surface area encouraging the colonization of microorganisms. They are characterized by a gradient of oxygen, water, different nutrient fluxes, and they are shaped according to the host genotype and the age. And both these systems, you have these microbiomes impacting the health as well as the development and growth, whether it is of the humans or it is of the plants. The perfect analogy that I'm mentioning is also, like I said, in terms of these gradations and how they impact the bacterial diversity. So if you compare these two systems, you will find a perfect analogy there. In fact, it is actually an inside-out system. So you will see when you move from the lumen to the epithelium, you have the highest microbial diversity inside compared to the outside to the epithelium. While in the case of the rhizosphere, the maximum diversity actually lies in the soil, which serves as a bank for the plant to recruit its microorganisms. So as you move inside, the microbial diversity is actually declining. What you see here, the black bars, are the different factors which affect the microbial communities. For example, the diet, the host genotype, the host immune system, and you have these factors also impacting the diversity in the plants. So like I said, it's inside out. What is most diverse and higher in number inside the lumen is actually outside the roots here. The factors which affect the microbiome, just like they did in the gut, also are impacting the rhizosphere, right? So whether it is the nutrient availability, whether they are abiotic factors like temperature, pH, the kind of soil that you have, different amendments. We were talking about pesticides and fertilizers and how that impacts the health of the organism. All these are impacting the microbiome. Now what happens is, the plant, it secretes different exudates, which makes this zone very rich, the zone near the roots very rich for microorganisms to colonize them, right? So the plant is actually dictating or recruiting its own microorganisms, the ones which are beneficial for them. And the microbiome, in turn, affects the growth of the plant in a positive manner. Now there is zoning. Just like I talked about macrozoning and microzoning, even in the plants you will have different kinds of microorganisms establishing or colonizing different parts of the plants. The maximum diversity, of course, is in the bulk soil, the soil which serves as a bank from which the plants recruit their microorganisms. This is followed by a very high diversity, and I'm talking about the fungal and the bacterial diversity in the rhizosphere zone, the zone around the roots. The differences in the zones is so much so that even if you have the same soil and you have two different plants grown in the same soil, because of something that I mentioned earlier, that is the root exudation, each plant will select a different community, whether it is below ground or above ground, right? So it is determined by the kind of exudation that the plant releases. Where do they come from? Just like in the case of human beings, you have a component, a small component, which comes vertically from the seed. But there is a huge component which is the horizontal transmission, which comes from the environment, whether it is the soil microbiota or the bioaerosols. Again, just like I talked about dysbiosis and in the animals, here also a good soil is categorized by a healthy microbiome, which leads to better functioning, better nutrient turnover, while a bad soil would have low yield, it will have more pathogens in them, they will be less in stress resilience. So these are the features of a dysbiotic microbiome, even in the case of plants. Now, because we have to replenish the dysbiotic microorganisms or dysbiotic ecosystems so that we have a good microbiome, there are several strategies that have been used in the plant microbiome management as well, starting from looking at the management practices. So we are trying to catalog different management practices and how that impacts the microbiome. Now we're looking at optimizing the practices for agroecosystem health. How can we actually manipulate the host? We know that the host genotype also impacts the microbiome. So can we really play around with this? Can we have improved cultivars wherein the plants itself select beneficial microorganisms? And these management strategies also revolve around the microorganisms. Like you saw the fecal microbiota transplant and the other therapeutics. For plants as well, you have different methods known for by which we can actually replenish the good microbiome. And what you see here are the first and the second generation. all this is leading to precision management of the microbiome and the plant microbiome. We work somewhere between the second generation and the third generation, and I work with all these approaches, whether it is management, the plant host, or even the microbiome-based strategies. Now talking about what I do in the case of with respect to plant microbiome, we try to engineer the rhizosphere microbiome, especially for climate resilient agriculture. Now, the microbiome-based solutions that we try to design is in 4 different crops. I'm sorry for this formatting issue. So we work with some of the legumes. We also work with wheat and tomato. And the kind of stresses that we work with include abiotic stresses. So we work with temperature, we work with salinity and drought. We also work with some of the pathogens, bacterial and fungal pathogens, and try to mitigate them in the plant systems. What we do differently is all the studies, the conventional studies, look at supplementation of individual microorganisms. For example, some of you might have heard of rhizobia, which leads to legume nodulation, right? What we do is we strategically bring together strains to increase resilience, right? Instead of sending one soldier to mitigate or to counter an army, we try to design the armies and send them at war. And then we also work with the microbiomes in toto. Without really dissecting the individual organisms, we try to design robust strategies wherein we work with the microbiome in toto. And we try to look at this triangle together. So we look at the plant, the environmental conditions, and the microbiomes, all of which are interdependent. What have we done? We've also done some conventional studies wherein we generate really good culture banks of the beneficial microorganisms. We try to understand the mechanism of action. So if you see, this is what a typical farmer would use. We have designed consortia and combinations which have been even visible here. You can see the kind of difference that we have resulted in. We've also started, like I said, bringing together strains. So we've designed consortia. We try to track them. Once you apply these consortia into the soil, one thing which has been very cumbersome is, can you really track them? When do you want to give a booster dose? So we've tried to do that as well under environmental conditions, real life conditions. We've tried to understand the mechanism of these synergism using metabolomic approaches. And we have also designed synthetic communities. And what I'm showing you here is a story wherein we were designing syncoms for mitigation of abiotic stresses. Then, like I said, we've also stepped up to working with the microbiomes in total, from synthetic communities to using the microbiomes. We are trying to map the country, several states that we have started off with in a very ambitious project by the ANRF and the DBT, wherein we are trying to see which soils are really suppressive to different pathogens, which have the inherent potential of being suppressive to different plant pathogens. And we have not just that, so this is when we are just surveilling, performing surveillance, we have tried to generate good microbiomes also. And the logic here is, if you expose a plant to a particular kind of stress for repeated cycles, what happens is the plant actually selects a microbiome which is tolerant to that particular stress. So this we have been doing for several generations, and we have been able to generate these good microbiomes. We have taken them to field, and what I'm showing you here is a farmer's field, something which was left barren. It was a saline field, and we took our strategies of using the microbiomes and converted them into actually arable land. Okay, so now, having told you about the human microbiome and the plant microbiome, what is it that brings them together? Microbiome has been considered as a unifying factor in One Health. Now, we've been hearing about what One Health is. Dr. Arul Raj was talking about it. The One Health is basically the philosophy wherein we consider the health of all components as one. So everything is interdependent, right? But what I'm showing you here is this triangle of One Health includes the animals, the humans, the environment, which includes the plant, and this is considered to be as One Health. We talked about the gut-brain axis. We are now talking about the soil-plant-human-gut microbiome axis. So these three ecosystems, or these three habitats, the human gut, the plant, and the soil. What you see here are different kinds of bacteria, what are listed here. We don't need to go into the details, but you see that there are differences, right? They are distinct ecosystems. Now, there are several microorganisms which are specialists, which you will find restricted to these different habitats, but there are also generalists. Generalists, by that I mean they are microorganisms which would be found in all these components. In fact, from the soil, the plant recruits the microorganism. These plants are ingested by the humans. So you see this direct link between the soil microbiome and the human health, right? So there's also a cross-kingdom microbiota which actually transfers or transmits between these different entities. This is actually called as a soil plant human microbiome axis, human gut microbiome axis. And this is where we stand. I know it's complex. This is where we stand when we talk about transmission of the microbiome. What I'm showing you here is the soil and plant, but you see there are several modes of transmission of this to the water habitats, to the wild animals, to basically humans and the air. And there are so many arrows that you see across all these components. What am I coming to? I'm coming to the unifying life, the microbiome in One Health. So now it's believed that it's actually the microbiome which is the common factor among all these entities. And there is a school of thought which thinks that instead of the three components that I showed you in the One Health, microbiome should also be involved or included as one of the components. What have we been doing in this aspect in the terms of One Health? We have been trying to perform surveillance of various human pathogens in the agroecosystems. We have actually been working towards developing strategies for minimizing the human pathogen load in the rhizosphere. This is this zone surrounding the roots. And subsequently, we want to minimize its transmission to the edible parts, which then are ingested by the humans. We are also working towards transmission of first performing surveillance and then performing mitigation of transmission of antimicrobial resistance through organic amendments in agriculture, which is being encouraged a lot. I hope I have been able to convince to you that microbiomes are a game changer for not just sustainable agriculture, but also for One Health. And these are the different SDGs that I mentioned can be directly linked to management of the microbiome, whether it is for nutrient balance or food security, for environmental sustainability, or even for food safety. And with that, I would like to start with the list of people I have to thank. I'll not name them, but these are just some of the collaborators that I've been working with in the country and also internationally. But I'm just the person doing the talking. There is actually a group of really enthusiastic PhD scholars and postdocs working with me. This is the current picture. This changes every 6 months, so there's a dynamics there as well. And I would like to end by saying that doctors save lives, soil scientists save the soil that sustains them. And with that, I would like to thank you all for listening. I would like to thank the administration for bringing me here. Thanks a lot.
Thank you, ma'am, for an inspiring lecture. Your insight into microbiome on healthcare and sustainable agriculture provided us with a deeper appreciation of how microbial communities can influence and contribute to healthier and more sustainable future. So, any queries from the audience?
Yeah, that was a very good talk, Dr. Sharma. You touched upon several aspects that forms one common ecology that takes us back to even the Indian philosophy, which says that all living and unliving and non-living organisms are the same. They come from the same 5 elements. That is on one side, but the question I have is that there's a group of soil scientists working on the mycelia, the mulch mycelia, which spreads miles, maybe hundreds of miles. There's a study in Miami, Florida, where they are studying this. It is believed that this is the factor that brings the water and the other elements together. That is one point. Is there any erosion of that? Or as one of the popular figures of Coimbatore has been working on in the Isha Foundation, he says that the topsoil that has gotten altered, and therefore all problems for humanity.
Yeah, so very well pointed. I mean, so by mycelia, I believe you're talking about the AMF, the arbuscular mycorrhiza, yes. Their importance has been well studied. I mean, it's well documented that they have a major role to play, and when I talk about microbiome, I include them as well. Regarding the topsoil, yes, it is true. It is the topsoil is believed to be the richest in microbial diversity, and because of several reasons. In fact, it's a catch-22 situation. Because of the soil being degraded, farmers apply more fertilizers, and because they apply more fertilizers, it is degrading all the more. So yes, this is true, and this is one of the scientific explanations that I'm trying to give here, trying to also supplement or replenish this topsoil by the microbiome strategies that we are talking of. I think it was all clear to all the students.
It is a very important topic about the microorganisms, how works. You have rightly compared the roots of the plants to the roots of the human being, roots of the human being. That gut is a human being's basic root for the nutrients of the body. Without the gut, you cannot live. Comparing the roots of the plants and the roots of the man, that is the roots is for a human being, the gut is a root, that is, you have rightly pointed out. But the importance of saving the topsoil with so many materials and also with so many fertilizers of various kinds, you may be sterilizing the topsoil without much organisms, madam. How you are going to save that soil from this type of chemicals from your side, madam? Your side means human side.
So thank you for pointing that out. In fact, one of the reasons why we have loss of diversity, one of the major factors, and I'll be very blunt here, is agriculture. Domestication of microorganisms. If you see wild plants versus the cultivated plants, there's an immense loss in diversity. So what now people are doing is more integrated. They're trying to see how they can use minimum quantity of chemicals together with supplementation of these more eco-friendly means, like the microorganisms, like the communities. That is very much being followed. And of course, like I said, it is not just the soil management through the microorganisms. We are also looking at designing plants and developing cultivars and plants which can recruit the good microorganisms from the bank. So there are several strategies in place, including the farming practices that are being played with.
The worst thing that we are all seeing is, madam, they are using some kind of chemicals to kill the plants, to kill the plants. They are also using so many chemicals, I don't know. Even my place, they will spray or something so that there will not be any weeds, there will not be any weeds. That type of chemical they are all using. We are worried about the soil microorganisms, madam.
I hope everybody now is bothered about it once I've shown you the connection between the soil microbiome and the human microbiome, and that awareness is important.
Thank you, madam. It's a wonderful talk, and I think the Tamil saying goes that katrathu kaiman alavu, which means that what you know is a handful of soil. You educated us more than a handful of soil to understand about it. So I've got two questions. One is that all of us come from an agriculture background. So in old age, we've all seen that there used to be the cattle in the field, and all the excreta from the cattle used to be there to enrich the soil, and we would plant the crops the next season. And the other thing what we saw is while you're doing rice harvest or sugarcane harvest, after the one or two thing, what we do is we'll plant a leguminous plant so that it enrich the soil. Does that hold true even in this modern science? That's the first one. The second one is antimicrobial resistance, which our microbiologists here see that often. I think we ran out of antibiotics. Is there any answers from the soil for this antimicrobial resistance?
Okay, so the first question first. Yes, these organic, these farming practices do help growing them, whether it is crop rotation or intercropping. They do help in actually replenishing or increasing the microbial load that is there, and that serves as a good bank for the next crop that is then planted. In terms of antimicrobial resistance, I have to tell you that less than 10% of microorganisms have been cultivated till now. So we are still to bioprospect the 90% or more which is there in the soil or in general, I mean in the environment. So what people are doing now, and that is called as culturomics, is trying to enhance this number of 10% so that we can get to the newer ones and try to see if there are solutions not just for antimicrobial resistance but for various other biotechnological applications as well. So we have just scratched the surface, I think.
Very nice, Dr. Professor Silpi Sharma. Thank you very much. Thank you very much. I now request Dr. Professor Chokkavelu from West Virginia.
Yeah, that was a very good talk. Dr. Sanjeev Chopra, who is brother of... it's okay, I can stand here and talk. Dr. Sanjeev Chopra, who is brother of Deepak Chopra, you all know, is one of the pioneers in Harvard University who is working on microbiome. And he gives a talk about microbiome, and he is a proponent of it. From medical standpoint, I mean, I can sit here and talk another hour or another 2 hours. The Tamil Nadu, where Dr. Parnisamy comes, he was trained in Stanley Medical College, and Stanley Medical College is doing a study on Pariyasoru. Pariyasoru is the good old days when people didn't have money, they didn't want to waste the rice, they put water in it, and the next day they eat the old rice. And this rice is a fermented rice, and they are showing study in the Stanley Medical College that this Pariyasoru protects you with good microbiome, and thereby doing irritable bowel syndrome treatment and Crohn's disease remission and ulcerative colitis remission, and exacerbations are reduced if you eat the Pariyasoru. It is good that Tamil Nadu, I just read in the January, Googled it, and of course it said in January of this year, the new minister Subramaniam is promoting Pariyasoru to people. That is very good. The second thing we can do, or the third thing we can do to improve the microbiome, is to eat plant-based food. The animal proteins are the detriment in the gut for the microbiome that are good. Plant-based food not only protects from heart attacks and protects from cholesterol, etc. The animal-based, they are also good for the microbiome. Third thing you can do is they have shown that the Mediterranean diet, again, kind of mostly plant-based diet, is cardioprotective, stroke protective, and vascular catastrophe protective. So these are, I mean, I can talk about this forever. And in the infectious disease field, we are pleading everybody to limit the antibiotic use to prevent the antibiotic resistance. And I used to tell people that people have vitamin deficiency, people have other deficiency, they don't have antibiotic deficiency, so you don't need to give them antibiotics. 9 out of 10 times they get better, and the IDSA is coming out with recommendation. We used to say 7 to 14 days for antibiotics. Now we are talking about 5 days or less, and 5 days and more for serious and less infection. So use less antibiotics, use shorter course of antibiotics. That is the take-home message. And the other thing I want to point out is that Clostridium difficile, which is a bacterial disease that is caused by use of antibiotics, is caused by the change of microbiome in the gut. And they have shown in people, particularly above 65, who get recurrent infections, by giving the fecal transplant, they can avoid their recurrent attacks. And they have also taken the bacteria, not only the fecal transplant, because you can also transmit other bacteria and the viruses. They have taken few microbiota and made them as a pill, and they are giving it for people who are getting recurrent C. difficile. And we funnily say, if you take the poop pill, you get you get better. So I stop there. I can talk forever. Thank you for giving me the opportunity to talk. Thank you, Dr. Palniswamy.
Thank you, sir. Every successful event is a result of collective effort and support. To propose the vote of thanks, I invite Dr. G. Velmurugan, scientist, KMCH Research Foundation.
Thank you all gathered here, and very big thanks, my first and foremost thanks to our chairman, Dr. Nallaji Palniswamy, Executive Director, Dr. Arun N. Palniswamy, for providing this platform, because it's very rare you can find this platform to discuss about all aspects of microbiome in a hospital. So first and foremost, a big thanks for that, and we promise that we will continue this culture, and we will create more things. And next, obviously, I thank the speaker, Professor Shilpi Sharma, just by a single email. She warmly accepted our invitation, and it's been a big honor. And also the way talk was delivered, right from the basics to the implementation, it created a lot of inspirations and new learning for everyone. Hope students has gained a big knowledge today. So big thanks to the Dr. NGP Arts and Science College, from principal, department HODs, and the students for actively participating in today's event. And also, this is the first time we have live streamed the Microbiome Day lecture. So through YouTube Live also, we had around 500 participants from different colleges all over the Tamil Nadu who has been listened to this lecture, and we are happy because our major aim is to disseminate the knowledge. So that's, I say, that's the fundamental right of a human being. So because we all grown up by getting knowledge from different people. So it's our duty to give it back. So that's a thing. So I also thank all the dignitaries here, and also a special thanks to Dr. Sokavelu for sharing his insights on microbiome from a medical perspective. Thank you, sir. I thank the Dean, Medical Director, Dr. Kalanidhi and Dr. Sridhar, sir, for joining this and gracing this event. And big thanks to Dr. Arulraj Ramakrishnan, Director of KMCH Research Foundation, who took all the efforts tirelessly to make this event sustainably continuing for years. And also I thank my colleague Dr. Mohan Raj for being a big supportive, and my research team. And I thank all the marketing team, photography support, and especially for this YouTube Live. Thank you one and all. Thank you. And last but not least, I thank the microbes which are present everywhere, which drives us. Because now studies shows even what we think, the way we speak, our aggression or our behavior are being guided by the microbes. So we thank the microbes also for this. Thank you all. Thank you.
With this, we conclude today's program. May I request everyone to kindly rise for the national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता। पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा। विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा। तव शुभ नामे जागे तव शुभ आशीष मांगे। गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता।